Good afternoon, everyone. Thank you for being with us on our Thursday afternoon webinar. Sorry for the delay, but we're happy to be joined by Dr. Mitesh Borad, who's a professor of medicine at the Mayo Clinic College of Medicine and Science. He serves as a program leader for the Gene and Virus Therapy program in the Mayo Clinic Comprehensive Cancer Center and is co-director of the Precision Cancer Therapeutics program at the Mayo Clinic Center for Individualized Medicine. Dr. Borad also chairs the International Cholangiocarcinoma Research Network and is a huge asset and ally for the Cholangiocarcinoma Foundation and for all the patients here. I'm Melinda Bikini. I'm the Director of Patient Services with the Cholangiocarcinoma Foundation. And we are going to be doing an audience poll real quick before Dr. Borad gets started on the webinar. So Rick, if you could launch that poll real quick, we will do that and then we will get going. And as Rick is launching the poll, I just want to remind everyone that you can put your questions in the Q&A chat or the chat, and we will get to them at the end of the presentation, and Dr. Borad can answer them. And again, I just like to remind everyone to try to keep the questions general and not to specific medical advice, because Dr. Borad won't be able to answer those specifically. But go ahead and feel free to put all your questions in the chat. And we'll give everyone a second to take the poll. Rick, you let me know how we're doing there.
Yep, it looks like just about everybody is answered.
All right, if you want to throw up the results, that would be awesome. And then we will get started. So that is what our audience looks like, Dr. Borad, if that helps at all. And I will throw it over to you and just let us know when to advance your slides.
Okay, thank you. Well, thank you, Melinda. It's always a pleasure. And I am honored to be here with everyone today and really excited to see how many participants we have. We don't even have this many participants on some of the lecture series we do for other researchers. So really happy to see all the engagement, and we are fully committed to working with our patient and research community to advance new therapies. So I will go over a study that has just started recently, which is the study with KIN-3248, which is a new Fgfr inhibitor. And then there's an ongoing study with another set of targets, which would be for KIN-2787, which is a RAF inhibitor. So really two study synopses here. Next slide. So the first one, as I said, this is a next generation fibroblast growth factor receptor inhibitor, mostly patients who have fusions of FGFR2, which will be about 7 to 10% of patients with cholangiocarcinoma, mostly the intrahepatic type that occurs in the primarily in the liver. And then there'll be also provision to allow patients who have mutations in that gene or amplifications in FGFR2, and also other alterations in FGFR3. And you see that commonly in bladder cancer, but there are some patients who have FGFR3 alterations. So nice to see that this study is a little bit broader from that perspective. So this is a phase 1 study. So first we have to find the dose. That's going to be the optimal dose. And then there'll be sort of a dose expansion at this recommended phase 2 dose. So for the first part, about 45 patients are going to be treated. Next slide. And I should mention also that patients who have previously received another FGFR inhibitor are also eligible for this part of the study. So again, this provides a nice option for patients who may have used one of the standard of care drugs or even a drug on another study to consider. In the dose expansion, there's a lot of focus on cholangiocarcinoma. That's cohort 1, as you can see. Then there's other patients with any kind of cancer, which is cohort 3, and then bladder and urothelial cancers, as I mentioned, which is cohort 2. So that whole expansion is about 75 patients. So all in all, about 120 over parts A and B. Next slide. So patients who would have failed maybe other treatments, maybe were even on another FGFR inhibitor, would be those that would be considered here. Again, it's not limited to cholangiocarcinoma, but this is a cancer where this target is very important. So it's a really big focus of this study. The drug is given orally, daily, without a break, as it's scheduled right now. But I guess as the study advances, the schedule could be modified, for example. But right now it's planned for daily dosing. And then the usual kind of blood and blood counts, chemistries would be done as assessments, and then scans would be done every couple of months, like is done in clinical practice and on other studies. There is a biopsy that's planned, as you can see here, and then there's some other biomarker tests that are planned to be in the study as well. Next slide. So the primary endpoint, because this is a phase 1 study, is safety. And then of course we know, because it's a highly relevant target, that we do expect it to see some level of effectiveness. So that is a co-primary endpoint. And then because this is a new drug, we would want to see how long it hangs around in the body. So that's a secondary endpoint as pharmacokinetics. And then we would look at other aspects of the drug, which would be all exploratory endpoints. Next slide. Eligibility criteria for this group here, generally have to have a good sort of functional status. The lab values have to be within a certain range. Tumors have to have FGFR2 or FGFR3 alterations of the various types I described. And then people are generally otherwise facing major health problems or ongoing issues. Those are listed as some of the exclusion criteria. But of course, people need to talk to the study teams to really get the sense as to whether this would be something they could pursue or not. Next slide. Going on to the second target, so this is KIN-2787. So this is a RAF inhibitor. We know these types of drugs can be effective in diseases, particularly like melanoma, where you have mutations in genes like NRAS or BRAF. So again, this is a phase 1 study. So you have to do a dose escalation to find the dose and study the safety. And then you do a dose expansion. Next slide. And then in the expansion cohorts, melanoma, as I said, is an area of interest. Lung cancer also has a reasonable frequency of these alterations. And we find these types of alterations also in cholangiocarcinoma, probably at a 3 to 5% range. So again, it's nice to have a study that's actively seeking patients across all different kinds of organ constraints, permitting all the other factors to use as a tool. Next slide. So again, this would be something in combination with another drug called binimetinib. Both drugs would be given twice a day orally. And then as the study advances, what doses those would be determined. Same thing, labs and scans are what would be done on the study. Next slide. Primary endpoint, again, because this is phase 1 study, is safety. And of course, because we know the target, we would look for some level of effectiveness too. So that's a co-primary endpoint, and then secondary and exploratory endpoints are similar to the other study I showed you. Next slide. So eligibility criteria would be, patients who have these alterations in BRAF, NRAS, the mutations that would fall into that RAF pathway, and these would be across various organ sort of constraints. So melanoma and lung cancer would certainly be ones that people would be looking for. But as I mentioned, for cholangiocarcinoma, it's 3 to 5% of patients who have these. And exclusion criteria would be, things that would make it difficult or unsafe for patients to participate, as I described earlier. Next slide. There is travel support for patients and their care team listed here. And because the study will only be open at a certain number of centers, this could be very valuable if patients find themselves residing in a place where it's not drivable, and that they have to fly to or establish temporary residence in to receive the therapy. Next slide. So I think that may be the last slide, and happy to take questions now.
All right, so I'm going to invite everyone to put questions in the chat, but I've got a few for you to start it off. Can you explain what it means, the difference between FGFR 1, 2, 3? Can people have more than one? Or, will they have FGFR 2 and 3? How does that work? Explain it to us.
Yeah. So FGFRs are a family of proteins, fibroblast growth factor receptors. There's 4 of them, FGFR 1 through 4, that are considered important. And in various cancers, you might find 1 or 2 or 3 or 4 that is the driver event, that gene got altered, and that's what's causing the cancer to go into overdrive. Usually you will only find just 1 of those. You won't find 1 and 2 in the same patient, or 2 and 3 in the same patient. It's going to be either 2 or 3. 1 and 4 are going to be very rare for cholangiocarcinoma. So we're really talking mostly 2 and then some 3.
Okay. And then for patients to find out if they have an FGFR mutation, will they be doing the testing during this trial, or will they have to know that going into the trial?
They will have to know that going into the trial. Luckily, this has become a much more standard thing to do. So every patient with cholangiocarcinoma should have genetic profiling of their tumor done, right? And of course, this message has been conveyed by the foundation very well over time, but just want to emphasize that again here.
Okay, I'm going to do a couple questions that came in. So if biomarker testing identified a BRAF D594G for an intrahepatic diagnosis, but they've already had gemcitabine, Abraxane, and durvalumab, would they qualify for this trial?
Yeah, I'll leave specific qualification issues to seeking out, the trial and making sure that other criteria are met. But broadly speaking, that sounds like a scenario where one would want to explore a trial like this.
Okay. For the FGFR trial, you stated patients may be eligible if they've been on previous FGFR inhibitors. Does this include if a patient has a known FGFR kinase domain mutation?
Yes, at this time. And this drug was designed to hopefully overcome many of these resistance mutations. So that would be particularly an area of interest in this study, to see if those patients can reacquire a response.
What would be any high-low blood chemistry tests that would preclude acceptance into the FGFR trial?
Yeah, again, I'll sort of leave specifics for when there would be formal engagement with investigators who are running these trials, but usually liver enzymes more than 5 times upper limit, or elevated creatinine values, elevated bilirubin values, platelets below 100,000. These are the kinds of factors that would likely be automatic constraints for participation.
Okay, how does one apply for the trial? I know that we can maybe find the link.
Well, don't feel like it's getting into college or anything like that. The trials are ultimately designed to hopefully help patients and at an early stage. So The easiest way to seek out where to and how to engage would be clinicaltrials.gov. You'll have a listing of all the sites where the trial is being conducted, and you can reach out to the center that's the easiest one to access for an individual person.
Emma, I don't know if you can, but if you can find that link and maybe put it in the chat where it has all the sites for the trial and how to find the trial. That way you guys can grab it. And if not, we'll make sure we find it and get it out to you as well.
Right. There'll be
Dr.
an individual link for each trial. Just keep that in mind, right? So.
Okay. We'll get that posted in the chat as well. Can you explain to patients on the call a little bit more about a phase 1 study, the dose escalation, and what a 3+3 study means?
Yeah. So when phase 1 trials are being done, that's the first time the drug is typically going into patients. So we don't really know what the right dose is. We don't know what all the side effects are. So that's the primary motive for the trial. So it shouldn't be confused that we already know everything and that we already know that there's a high likelihood of it working. We don't in phase 1 trials. With that being said, for very targeted therapies, there's still a chance that one could benefit as a patient, even though it's a safety study. 3+3 means we evaluate 3 patients at a time, make sure they do okay before we go to the next dose level. If there's issues with side effects, then 3 more patients may be enrolled, and you can either go higher or stay where you're at or go lower depending on what you observe. So that's generally speaking what the 3+3 means.
And will you explain a little bit about Rhesus criteria?
Sure. So Rhesus stands for Response Evaluation Criteria in Solid Tumors. It was basically a way to standardize how the measurements of the tumor are done and when to call it a response, or what the term people use, stable disease, where there's not much change, versus it worsening, called progressive disease. So it's you make these measurements, and if there's a 30% difference, it's a response in the shrinkage direction, of course. If there's more than a 20% difference, that's progressive disease in the other direction, obviously. Or if there's new spots, that's considered progressive disease. So these are just criteria doctors can use no matter where you are in the world to standardize things.
Okay. Would you touch a little bit more on resistance criteria and what that means? Not resistance criteria, resistance mechanisms with FGFR.
Yeah, we don't know everything yet about why and how and when this resistance occurs. Some patients don't respond to begin with. We don't understand fully well that how that happens, for example. We do know some where patients have been on these inhibitors and clones or cells that have a mutation which doesn't allow the drug to bind and have its effect, can cause resistance. So these are terms like gatekeeper mutation, et cetera, that you might find in the literature. So that's what that refers to. So generally the strategy is going to be design a drug that binds in another site, not the gatekeeper, and you can hopefully overcome the resistance. But it remains an area where we need to continue to understand why resistance occurs and what could be ways to get around it.
Okay, there's a couple questions in here about what side effects should we expect with this study. Are they similar to the current FDA approved FGFR drugs?
Yeah, broadly speaking, that's what we would anticipate, that there would be a class effect. So effects on the eyes, the skin, the nails, elevation in phosphate, some fatigue, some hair loss, things of this nature. But because it's a new drug, we may see some unique side effects, and that is one of the purposes of the study. For the RAF inhibitor, again, there have been drugs in those categories, and usually you expect things like skin issues when given by themselves. Sometimes they cause some abnormalities in the skin, both tumorous and non-tumorous. When given with that other drug I mentioned, those are minimized. And sometimes those drugs also caused eye issues amongst other effects. So some class effects, and then of course we'll be looking for some new unique things that could be specific to the drug.
And we've heard the terms first generation FGFR inhibitor, second generation. Can you explain what that is?
Yeah, and this is not a standardized nomenclature yet, but first generation we could say maybe are the reversible, ATP pocket inhibitors, for example. The second generation could be the irreversible ones, so drugs like futibatinib, which came there after infigratinib and pemigatinib, for example. And third generation is a broad, not well-defined term yet, but that could be drugs like the one I showed you, or an allosteric inhibitor that binds to a different site than the ATP pocket called RLY4008. And I'm sure there'll be a fourth generation as we move along. So it's quite amazing that we're already into the third generation.
So to me, that just sounds like the science keeps advancing.
Exactly. The science is advancing at a very rapid pace, and it's challenging for everyone to keep up with things, let alone patients, even patients, physicians, and researchers are finding it challenging to make sure that they're staying one step ahead, hopefully, but if not, just in tandem.
Okay, so was 3248 developed in a way with the goal of dealing with any FGFR resistance mechanisms that other approved drugs in development showing promise are not addressing? So are these maybe the mechanism of action different from others?
Correct. So drugs that fall within this third generation would need to have that, because there's already drugs that can be given otherwise, so there wouldn't really be an appeal for anyone to evaluate drugs that don't tackle the mechanisms of resistance. So that was the intent, but we'll have to see how it pans out as we take it into the clinic.
So then what happens after the phase 1 study is fully accrued?
I would hope that there would be some interesting signal there once that's done, and that there would be enthusiasm from everyone, from the biopharma community, patients, the advocacy community, researchers, to continue to develop the drug into phase 2 and beyond. But we're in early days, so it's difficult to speculate.
Okay. Any other questions? I'll give a few minutes for anyone to throw any more questions in there. And in the meantime, I just want to emphasize how grateful I am that they are providing financial assistance to patients who enroll in this study. That helps us all to have access to be able to participate. So that's great. I don't know. I think if you have any questions on that, when you find the number on the clinical trial, you can call and ask the study coordinator and they can give you information on that, but that's very beneficial. I do not see any other questions. I guess, so if you're testing for mutations, is there a difference between testing between BRAF and FGFR? Does it have to be tissue? Can it be blood? What are they looking for?
Yeah, nowadays people are exploring both because it's easy to get a test off of blood, but you could miss certain mutations sometimes, particularly the fusions on the blood test with what we have currently available. So I would encourage patients to have both tested if they can. Of course, there could be individual insurance constraints, or just differing practices with physicians at this time. But both avenues are fair game for finding these events.
What is considered a creatinine that is too high?
This depends on every study. Many studies have a creatinine of 1.5 times upper limit of normal being the cutoff. Some have a glomerular filtration rate that they will use, and they will use 50 as the number. But again, these kind of nuances would be things to check with the study teams if people are interested in pursuing these efforts.
Okay, so can we post the BRAF clinical trial number in the chat as well? And if we don't get it in the chat, we will make sure to add it to the recording of the webinar when we send it out. And we will also make sure they are posted to the Facebook groups for patients and caregivers so that they know where to go to look for questions. Dr. Borad, do you have anything else you'd like to address in regards to the clinical trials?
I think the gamut of questions really covered the key things, which would be the resistance, is there support for the trial if you have to travel, how do you find these events?
And these both are open right now and accruing, is that correct?
Yeah, not at all the centers that have been planned, but over time it will be available at all the centers that are in the process of opening. So some already, and some soon to come, and some come later.
Okay, and we should have that information up on the website as well as the clinical trial posted on our clinical trials page. And it should list the sites and have the contact numbers there. So we'll make sure we get that information out. Thank you everyone for joining us. And if you have any questions, please reach out to advocacy@curecca.org. And we're happy to answer any questions. Thank you, Dr. Borad, for the time you took to explain these trials. We appreciate it very, very much.
Absolutely. Thank you for having me here today. Bye-bye.
Bye-bye.